I'm good.
Nice to see you. We get
Yeah, good to see you.
appointment every year.
Yeah, exactly. It's a good tradition.
Yes, yes. Okay, great. So I'm delighted to yeah, maybe I should look here. Oh, I'm delighted to greet David Unhoyes, who gives this very nice lecture about clearing and expansion microscopy every year. And so nowadays you're working at iLife or you're working at KTH?
I'm at Silent Lab, and then we also have this startup company now that I'm there at 30%.
Okay,
But I'm
great.
yeah, I'm located at Silent Lab. Yeah.
Thank you.
So should
Please,
I
yeah.
start?
Yeah.
Yeah, let's see.
And for everyone, I've just uploaded the PDF, so if you do not see it, just refresh your browser.
So this is all good now. You see everything?
Yes, yes. Perfect. Thank you.
Yeah, good. So thanks again, Sylvie, for the invitation to present here at this nice course. So I will give kind of a brief overview of different clearing protocols and also some expansion protocols. Probably most of you have heard about these. They are not so new, these techniques anymore. They've been around for a decade or so. But I will try and give an overview of my experience working with these protocols and hopefully be able to give you some tips and tricks. So I work in the kidney field, so I work mostly with kidney samples. So what you see here, wait, I will just change to this laser pointer. Yeah. So what you see here is this is a quite thick piece of mouse kidney. It's yeah, 4 or 5 millimeters or so, which is here cleared. And here you see an expanded kidney sample. And these protocols, as you will see, they are kind of related to each other. So yeah, let's just jump into it. So you have now heard a lot about uh, how objectives work, I guess, and how we are using objectives, lenses to focus light into a sample. And what happens in biological tissue when we are trying to do this deeper inside the sample is that we will have scattering. And this scattering occurs because we have tiny differences in refractive index as we are going through uh, the tissue. And when a photon hits one of these changes, it might get scattered. And this probability, of course, increases the deeper we go inside the tissue. So this is what it would look like in my case, in a kidney sample. Here we have stained for the filtration units in the kidney, the glomeruli. So at 10 micrometers depth, so here we are still pretty superficial. We can acquire nice images, but as we go deeper, we get more and more blind. And at 1 millimeter, we basically only have noise left in the image. And optical clearing protocols, they were invented to deal with this issue. So the first description of an optical clearing protocol was actually in Sweden in 1905. So this guy, Halvar Lundvall, he played around with immersing human embryos in different solvents and different oils. And he found that by doing this, he could render them transparent. So this was more of a proof of concept publication. And then a few years later, a German guy called Spalteholz, he described this also from a more theoretical point of view, why this happens. So he usually gets the credit for to be the inventor of optical clearing. And optical clearing, what all of these protocols aim at doing is to homogenize the refractive index throughout the sample. So we don't have these jumps in refractive index where photons can get scattered. And there are different ways of doing this. So you have the different constituents of the tissue have different refractive indices. So lipids and proteins, they differ slightly. So you will have scattering at these interfaces. so you can get rid of lipids, which is what most protocols do. Also, the mean refractive index of the tissue itself is then in this range here. So if you immerse your sample in water or PBS, since you have this refractive index jump between the environment and the sample, this will render the sample opaque. So you can immerse your tissue in a solution with high refractive index. There's also another way, so you can make sure that as much of your sample as possible is of the same substance, in this case, water, so you can hyperhydrate your sample. And yeah, all the optical clearing protocols out there, they do one or a combination of the above. So this is from a review. It's now 10 years old, but still you divide these optical clearing protocols into families in the same way as presented here. So you have these different protocol families, so to speak. And the first one is solvent-based clearing. So as the name implies, you then immerse your sample into a clearing solvent. So then you first have to dehydrate your sample, and this can be done either in methanol or tetrahydrofuran. And then after this, you immerse your sample in the solvent, which is all of these solvents are of high refractive index, and they also, to some extent, remove lipids from the sample. So in this way, you render your sample transparent. Then there are the simple immersion protocols. So these are aqueous solutions with high refractive indices. And these are, I would say, the simplest clearing protocols. So here you take your sample, you don't do anything about the lipid composition, but you just match the surrounding refractive index. You try to match it to the refractive index of the sample, which will then increase the transparency. And then we have these hyperhydration protocols. And here you use urea, for example, to drive water into the smallest compartments of your sample to try and make sure that as much of your sample as possible is water molecules. And this will also homogenize the refractive index in the tissue. And then lastly, we have the hydrogel embedding protocols. So here you would introduce hydrogel, which will then more rigidly fix your sample. And after this, you can use a harsh detergent such as SDS to delipidate your sample. So you use high temperature, you use SDS in a high temperature, and you can also even apply an electric field over your sample to facilitate this delipidation. And after that, you, as a last step, you immerse your sample in one of these aqueous solutions. And then you have rendered your sample transparent. By the way, if there's anything unclear as I'm moving along, just interrupt me at any time.
Yeah, I have a question.
Yes, go for it.
Okay. Why using methanol or THF? What is the difference with, for example, methanol or ethanol?
I think for some protocols, so I haven't used solvent-based protocols extensively, so I only have experience with methanol. I would guess you can use ethanol too. And I cannot actually I cannot comment on the pros and cons of using ethanol as opposed to THF or
Yeah,
methanol. I don't know if you, Sylvie, have any.
No, I don't know really, because there are so many different protocols, so I don't know of anyone who has actually compared. I think the best way is probably to go to the article and they probably compare there.
Yeah, because so far that I know, the methanol, ethanol, there's a part where in the solvent clear, well, the solvent protocol that it's to bleach the sample and then to do the rehydrate and do the staining, the dehydrate. But what is so specific about methanol, or for example, to see with ethanol, is it like different, or because both are alcohol? So that was my question.
Yeah, I mean they are also probably, I'm just guessing that they will also denature proteins to different extents, which can be negative for some applications. I don't know if you have fluorescent proteins there or so. But yeah, I'm not the expert on solvent-based protocols, so I don't really dare
There's
to.
a huge difference between methanol and ethanol. Methanol if it destroys completely, for example, the cytoskeleton, and ethanol doesn't. So they behave in a very different way. It's not just about are they alcohol or not.
Okay, okay, that's good to know. Thank you.
Yeah, so there are, I mean, there is now over the last decade there has been an explosion of different clearing protocols. Yeah, so there's a whole ocean of different protocols to choose from. So I think this can be a little tricky if you have no experience at all with this to know where to start and which protocol to choose. So I just made this little list of the different considerations that you would have to make. So I think the first one is what degree of transparency is actually needed for your scientific question, because usually the more transparent you need your sample, the more complicated your protocol is going to be. So if you can work with, say, 300 micrometer thick sections or so, then probably one of these simple immersion protocols are sufficient. If you need to image a whole brain, yeah, then you would have to go for the more complex protocols. Then also your labeling strategy is important. So if you have fluorescent proteins in there, some protocols are beneficial. Some of them will quench all the fluorescent proteins. So then you would have to stain for them to make it work. And also, I think it's pretty important to consider what imaging modality do you have access to. And there, especially the refractive index of the optics is the important part. So for instance, like all of these different clearing protocols, they have a final refractive index of between, yeah, almost water, so 1.35 or so, up to 1.57. And when you are imaging these large samples, it's really, I mean, you really have to consider that you have matched, that all the optics is perfectly matched. So I think this is very important also to consider. Yeah, so you have to find a protocol which works with your labeling, transparent enough for your sample or your scientific question, and compatible with the imaging method. So this is basically just because when I started my master thesis in 2013, I started to use hydrogel-based optical clearing, and I almost only have experience using this sort of subset of protocols. So that's why I will now dive a little deeper into this type of clearing. I will also, yeah, give you an overview of the protocol that I use. I'm not saying that this is the best protocol. It's just because it's the protocol I know. So the idea of hydrogel-based optical clearing is depicted here. So the first thing you do is that you fix your sample with PFA, and either while you are fixing or afterwards, you make sure to introduce acrylamide monomers into your sample. And when you polymerize these acrylamide monomers, yeah, you have this hydrogel matrix that is bound to all the different molecules within your sample that has amine groups, so proteins typically. And this creates a much more rigid fixation of your sample. That's sort of the practical implication of introducing this matrix, which then allows for dissolving lipids using these very harsh detergents at high temperatures without risking to compromise the morphology of your sample, the integrity of your sample. So after removing the lipids, you are left with, ideally, you're left with only your molecules of interest, the proteins bound to this gel. And then as a last step, you then mount your sample in a high refractive index aqueous solution to render your tissue transparent. And these are just a couple of examples. It's kidney tissue, of course. So this is a fourth of an adult kidney. And this has fluorescent protein expression in the filtration units of the kidney, so the glomeruli. This is a late embryonic rat kidney, and this is stained with wheat germ agglutinin, so this will visualize the tubules in the kidney. And these samples are both around half a centimeter in size.
I have another question, sorry. Really nice images. I just wanted to ask now, because you mentioned that with the you get the polymers into the cells. Do you see any changes in size by doing this protocol?
Yeah. So you mean the size of the sample itself?
Yeah, the
If it,
sample itself,
yeah,
because you are inserting this. Does it get suppose bigger?
yeah. So this is it's a good point. So these protocols that I use, they will all swell your sample a little bit. So this is anywhere between 20% to 50%. And this is good for some applications. So I mean, you will see this later. I mostly do high resolution or super resolution imaging, and then you actually get a little help from this. So you get a little resolution improvement from this swelling of your sample.
So there should be like during the analysis a readjustment of these sizes or something like that, because if you have this expanded tissue, you have to readjust your analysis because it's a bit different from
Yeah,
the true size. Okay, thank
absolutely.
you.
So
Yeah, yeah, yeah.
if you're interested in absolute numbers, then of course you have to adjust for the expansion factor, the swelling factor.
Thanks.
Okay, so then I will just tell you a little bit about my protocol that I use. So this protocol is based on clarity, as I already said. So when I did my master thesis in 2013, my task was to optimize this clarity protocol, or to set up this clarity protocol to apply it to kidney tissue. And the first protocol was extremely complex, especially it involved applying this electrical field over your sample. And then you had to pump SDS with a pump. You had to use liters of SDS. It was very, very complex just to win a little bit of time so that the clearing was, a little bit faster. But most people, they dropped this immediately, this electrophoretic clearing step. So most people just started to do the passive clearing, which is just shaking at high temperature. And then also, uh, this paper was published in 2017, where they showed that actually this also works without introducing the hydrogel at all. And this is because the tissue itself is actually acting as a hydrogel. So you can actually omit this introducing this gel completely. What should be considered then is that you have a less rigidly fixed sample. So for very large samples, it might be that if you clear them for a long time, that you might compromise the morphology of your sample. But we found out that for our application, it works almost always without introducing a gel at all. And the protocol that we use now is published in 2021. So you can go and read this if you're interested in the details of the protocol. So this is the overview of the protocol that I would start using for any application, not only for my own, but if I collaborate with someone and I start, I just start from this protocol always. So it's just fixing the sample in PFA. If you need to, you section the sample. So here, I typically, for my applications, I would section at anywhere between 200 and 500 micrometers, which is good for what I do. You then delipidate your sample in 4% SDS, and this you do anywhere from 1 to 72 hours at 50 to 80 degrees, and this depends on the sample size and also a lot on the sample type. So, for instance, kidney is very different from brain. So this you would have to optimize for your application. And then prior to imaging your sample, you would then embed it in this aqueous high refractive index solution. So we use fructose, but this is mostly because it works well for our super resolution imaging. Otherwise, we would use Histodense or Omnipaque. So these are good options for maximum transparency. So that was just that was just to show you that this protocol now it's not a it was sort of optimized for simplicity, so it's not a complex protocol. I would say it's one of the simpler clearing protocols that you could apply. So it's anywhere between 5 to 80 hours of sample prep, and this depends on also on the sample size and type. And if you don't have any labeling, you don't have any fluorescence in the sample, you don't have any fluorescent proteins, you would have to stain your sample, and this you do between the delipidation and embedding step here. And this would also add, for very large samples, it might add several days of antibody incubations. So just a little summary. I would say keep it as simple as possible. It's always complex enough to clear samples, and you will do a lot of troubleshooting. So start as simple as you can. We have good experience with the following protocols. For aqueous, it's this protocol that I just presented to you. Cubic is also a good protocol. It's also reasonably simple. Especially you can simplify this protocol even more than the published version.
I just have a question. What you call fast clarity, is this the protocol you have published in 21?
Exactly. That's the one I just presented here in the preceding slides.
Thanks.
Yeah. And as I said, for solvent-based clearing, I don't have much experience. I have used ethyl cinnamate, which is a nice protocol because it's extremely simple. It's just there you use methanol to dehydrate the sample. You put it in ethyl cinnamate, which is a completely non-toxic solvent. So there are other protocols like iDisco, which I haven't applied, but a lot of people are working with this protocol, which is a very good protocol transparency-wise, but it contains some toxic reagents. So this can be an issue. It can also be, I think it can be corrosive to some optical elements, so you have to be a bit careful there. With your
No,
imaging.
this is a question in regards of your experience with your protocol, because the one criticism that I've heard about the iDisco is that you have to look for a list of tested antibodies that work or don't work with this solvent based method. But so far in your experience with the Clarity hydrogel based protocols, what is the percentage of antibodies that normally work in normal staining, but then you go and do this really nice application with hydrogel? What's the percentage of they working in the normal tissue and with the Clarity tissue?
Yeah, that's a very good question. So it seems like if they work with PFA fixation and paraffin embedded tissue, they normally work with this hydrogel approach. If you have an antibody that only works in like fresh frozen sections, for instance, it usually, I would say, doesn't work. So I mean, it's based on PFA fixation, and it's very hard to get rid of this, I think, with these protocols. So that's my experience. So if it works with paraffin, PFA, then usually it works
But
also
not,
after curing.
but PFA frozen, not so much.
If your antibody only works with either fresh frozen
Hmm.
or say it only works with acetone fixation and it doesn't work with PFA fixation or paraffin or anything like this, it probably won't work.
Okay.
Yeah. So
Thank you
that's
very
my
much.
experience.
Yeah,
Yeah.
very, very interesting. Thank you.
Yeah, and then a little bit about the labeling of these samples. So, of course, if you have endogenous fluorescence, this is, I would say, highly recommended because staining cleared samples, especially if you want to stain large samples, it's always going to be challenging, and you're going to consume a lot of antibodies. So yeah, fluorescent proteins always the best, but the clearing protocol has to be compatible, as I mentioned before. So some solvent-based protocols will quench some fluorescent proteins. There are solutions to this also on the solvent-based protocols, that there are some that are compatible, but this is one thing to consider. So, and if you're labeling with antibodies or dyes, of course, the probes have to penetrate these thick samples, and you would have to use very high concentrations of the antibodies because it's not only the diffusion itself, it's also that your epitopes on the surface of the sample will consume all the antibody before it had the time to diffuse into the sample. So that's why you really need to crank up the concentration in order to fast saturate the epitopes on the surface to facilitate diffusion of unbound antibodies. Yeah, and that's why it only works with very, very good antibodies because you need to use these very high concentrations. And in my experience, the limiting factor of the success of the project is actually the labeling. So most of these protocols, they will render your sample transparent, and if you have chosen the right imaging modality, it will work, but the labeling is usually what's problematic.
Uh,
Okay.
and in regards of the concentration, by your experience, when you say that the concentration has to be high, but you have one concentration that works in normal histology. What, by your experience, how many times higher? Like twice, thrice, the concentration?
Yeah, a factor of 10, I would say, is
10. So you will go by 10. So
yeah,
it's 1,100.
exactly. Yeah.
Okay.
So
Okay. Thank you.
that's a good starting point at least. But then it really depends on the if you have a very, very sort of highly expressed protein that you're staining for,
Mhm.
you might have to go even higher because you have so much protein that it will just consume your antibody
Mm,
right away.
okay. Great. Thanks.
Spoke a tiny bit about the switch protocol,
Yeah.
and you have been using this as well. Maybe you speak about it later.
No, I have used it. We tried it. It's yeah, we didn't use it with good success actually.
Okay.
We didn't really it worked for, but it has to be optimized for each labeling, each antibody. It seems like you need different concentrations of the SDS, which is what you use to sort of inhibit the binding. And in our hands it was just too sort of hit and miss with the different staining, so we don't use it typically. But I think now actually uh, the people who published it, they have a long list of different antibodies which they have validated with this protocol, and they also give the different concentrations that they use, I think, for the SDS. So it might be now that it's yeah, if your antibody is on this list, it might be easier. But I spoke to other people who tried to use Switch, and they had the same experience that it was much harder than you would get the impression of when reading the paper. Yeah.
Interesting, because we have some users who have used it and it worked fine, but maybe it was just luck.
Yeah, or maybe I'm a bad Switch user. I don't know. But that was my experience at least. Okay, so then that was everything I had to say about clearing, and now we move into tissue expansion. Also here, I think most of you have heard about these protocols. They are similar to these hydrogel-based clearing protocols in the sense that you also introduce a gel. But here the difference is that you also include sodium acrylate in this gel. And if you polymerize sodium acrylate in a salt-rich environment, you get these very curled up polymers, which when you put them in DI water in a salt-free environment, they will straighten up like this, absorb a lot of water and straighten up. And then in the ideal world, you have now moved all your biomolecules that are bound to this gel isotropically further apart from each other. And that's then how you gain the resolution in this case. So it's a completely non-optical method of increasing your effective resolution. So the idea is very simple, but the problem that you face in biological samples is that even though you have embedded them in this gel, the forces within the sample will inhibit the expansion. So this, if you have a big piece of tissue in your hands, you cannot sort of pull it apart. So you have to do something about these forces that are inhibiting the expansion. And then Boyden published in 2015, so this was the first solution, that you can use these oligomer conjugated antibodies. So you stain for your target of interest in the biological sample. And then on each antibody, you have these oligomers that are conjugated to the antibody and also to the fluorescent dye. And then you can link these oligomers to the gel that you introduce, and then you just use enzyme to digest away basically the whole sample. So what you are left with is this gel with a fluorescent map of where your target used to be before you got rid of it, if that makes sense. And then you can, of course, freely expand this gel because now there's no biological tissue here anymore to inhibit this expansion. And you can then in this protocol, you could achieve around 4 times expansion, so 4 times resolution improvement. Of course, these protocols, they relied on these custom-made antibodies. So there was also another solution published a year later, which is called ProEXM, so protein retention expansion microscopy. And here you can use conventional antibodies. So you just stain your sample with these conventional antibodies, and then you introduce a linking group, which will link small fragments of the antibody and also other proteins in the tissue to the gel. And then you use an enzyme to cleave all of these proteins and partly digest your sample. So now this will allow the gel to expand more freely. And this also gives you a similar expansion factor. Then there's a third solution called MAP. And here you don't digest proteins, you denature the sample instead. And this protocol is very, very similar to the Clarity protocol. Actually, you introduce this gel, only now it contains sodium acrylate, and then you denature in SDS at high temperatures. So it's very similar to this delipidation step. And after this, when putting the sample in DI water, now since you have denatured all the proteins, it will more freely expand. Yeah, and they show that this works for most tissue types. And this I will show you later. We have applied this to kidney tissue. There's also a fourth solution called cubic X, and this is hydrogel free expansion. So here again, you're taking advantage of the fact that the biological tissue itself acts as an hydrogel. And then they screened hundreds of different chemicals, and they found an optimal chemical, which then allows for a 2 times expansion of your sample without introducing any hydrogel at all. So yeah, these protocols, they have all been applied to different samples, and yeah, they work well. They work differently well for different applications. And this is just a short summary of these protocols. So the original protocols, I mean, it had this con that you have to use the oligoconjugated antibodies. So I don't think this protocol has been applied that much. This is a very popular protocol, ProEXM, because it works with conventional antibodies. And here you are staining your sample before expansion. So you are just fixing your sample as usual, staining it on a slide or so, and then you continue with the protocol. And this can sometimes be beneficial. The MAP protocol, on the other hand, it also works with conventional antibodies, but here you are staining after you have expanded your sample, after this denaturation step. And for some antibodies, this has been shown to be beneficial. And then the cubic X approach, it works also, of course, with the conventional antibodies, and it has the advantage that you don't have to use any polymer. It's then less toxic and it's much simpler, but you are compromising resolution since you have your the lower expansion factor. And then more comments. I mean, I have some experience with the expansion microscopy, and this can be very powerful. Of course, it's very intriguing that you can now increase your resolution without doing anything on the optical side. But it should be said that the protocols are often quite laborious, and practically it can be demanding to work with these samples. Especially, I have applied it on tissue samples, and there it's especially demanding, I would say. So you have DI water embedding, and this can give some drift issues because the sample is very wobbly. It's just this gel floating around in water, so it can be hard to keep it completely still. It can, for tissue samples especially, it can be hard to mount the sample close to the cover slip, because now if you want to use a high NA oil objective, your working distance is usually quite limited. And then if you have some uneven parts on the surface of your sample, you will expand also this. And it can actually be hard to even find pieces of the sample that are close enough to the cover slip. So I would only apply this if you really need this higher resolution to answer your question. Sometimes super resolution microscopy, if you have access to it, can be a simpler option. I should also say that there are protocols now. I mean, they have developed these techniques, so there are protocols that achieve an expansion factor of up to 20 times, maybe even more now. But yeah, then of course the complexity increases even more. Yeah, so I think that's all I had about expansion microscopy. I think I'm running a little late. Am I, Sylvie?
See. Yes, I think so. A little bit. No, actually, no, not at all. You have until 25, so no problem.
Yeah, okay. Good. So I think that's sort of everything for the expansion part. I don't know if you have any questions so far.
I just want to mention, I don't know if you have seen, David, the paper that came out of KI, Perlin, last year, about clearing of whole brain that is compatible with RNA labeling.
So it, yeah,
Pretty exciting.
Yeah, that's pretty cool. So
I put it in
I
the
have no experience.
Okay, so I put the link in the lecture if you want.
Yeah, okay. Yeah, cool. So no questions about expansion? Does
Anyone
anyone
tried?
have experience?
So, do you think that expansion is a bit of a niche, or do you think this is something that is going to develop?
Well, I think it's going to develop. For some applications, it's extremely powerful, and it's um, one good thing is that, of course, you are isotropically expanding your samples. You're also getting this higher SED resolution. And this, for some applications, can be really good to if you have very crowded sort of structures that you want to image. This can really be a limiting factor for some super resolution modalities, such as STED. It doesn't work very well if you have a very packed structure. And there it can really help to dilute your signal also in the SED dimension. And I think also these drift issues, there are more and more solutions coming up dealing with them. So I think it's getting easier to use those. So I think, but it's for very niched scientific problems, I would say, that fall within this, I don't know, yeah, 50 to 150 nanometer range where, yeah, you can not access them otherwise. And so then I mean, I have only a few more minutes, so I will not, this is more about how I apply these protocols. So we are looking at the kidney, and we are looking mostly at the kidney filter. And these are tiny structures. So the width of these foot processes, it's 150 to 500 nanometers. And the junction between the foot processes is only 50 nanometers or so wide. And what we have shown is that we can resolve these structures now with optical microscopy. So these structures could only be resolved before with electron microscopy. And also in the clinic today, you apply electron microscopy to visualize these structures. But we showed what can be a little interesting to say here is that it was only when we combined optical clearing with super resolution microscopy that we could achieve a good enough staining quality and a good enough contrast to be able to do this. So here, for some targets, it can really be beneficial to clear the sample also just to increase the quality of the staining. So this
I
is
just
maybe
wondering, David, sorry, Sophia, you had a question, I think.
Yeah. Go ahead.
I was wondering, when you are looking for something within tissue and then you expand it, is it hard for you to then place it sort of within the frame of the whole tissue? So we learned when you have high NA objectives, you only have small field of view. Does it mean that then you have to tile, or how do you put it in context of the whole thing?
I think that's a good point that you are, of course, your field of view is then shrinked also by a factor of 5. So if you want to catch a whole large structure, you would have to tile. But I think when applying expansion microscopy, you would only apply it to smaller fields of view where you have sort of a high resolution scientific question. So it's a very bad method if you want to image large samples, of course, because it will take you, yeah, much longer.
Thank you.
And it's 5 times linear expansion, so it will actually, it will take you 25 times longer.
Thank you.
Yeah. So I think this is maybe interesting to show that also you can, for some targets, you can really increase the labeling quality using these protocols. So that's the main reason actually that we apply them for our work. Yeah, and this we published in 2015. Yeah, I would just rush through these last slides. So we are now also, what we are doing is we are quantifying changes to the kidney filter.
I think somehow you stopped sharing. Maybe it was on purpose.
I stopped sharing. No, it says I'm sharing still, but then
Maybe it's just me not seeing it.
No, we can see
wait.
the screen. We are fine.
Didn't say
Okay.
anything.
Yeah, so we are, I mean, we have focused on quantifying. So we have developed a lot of tools to, from these images, extract meaningful quantitative information that describes pathological changes to these tiny structures. Yeah, and you can read our paper from 2020 if you're interested to see. So this led to a new model for how kidney filtration works and why it stops working when you have these pathological changes to the filter. We have also applied a lot of deep learning-based segmentation. So we have trained a deep learning network into segmenting these structures completely automatically, which then, of course, increases the data throughput of these methods a lot now. And we have also applied tissue expansion to visualize these structures. And we combined it also with STED microscopy in this paper from 2018. So you can read this paper if you're interested in the details of this. Yeah, so that was basically everything that I had. Also, I'm not part of the ALM facility at SciLife Lab, but I work a lot with these microscopes, and I just wanted to say that, especially when it comes to super resolution microscopy, we have a lot of experience and we can support projects. So yeah, just reach out if you need help with this. So that's all I had, and send me an email if you have any more questions.
Thank you, David. So we will hear Hans next week.
Yeah, exactly.
Yeah. Are there any more